Hi, my name is David Ramirez. I am Associated Scientist of the International Potato Center and the head of the crop ecophysiology and modeling laboratory of this institution. Today, I will present a study that assesses the usefulness of carbon-13 discrimination as a tool for the analysis in an integrative way of the physiological performance of potato under different watering conditions. The latest IPCC report predicts the alternate occurrence of pulses of torrential precipitation and severely dry heat waves in potato producing areas. This extremely unstable climate scenario requires resilient potato genotypes with highly adapted to different soil-water scenarios. That adaptability will be the result of the integration of different plant traits that must be assessed for selection towards productive germplants. For example, under mild water restriction, increased photosynthesis rate, stomatal conductance, vegetative growth will be desirable for taking advantage of water pulses. However, during terminal severe drought, stomatal closure and water use efficiency are more important. Carbon isotope composition is an integrative plant trait which depends on different thermodynamic and kinetic processes during crop growth and development. This trait is measured as the ratio of carbon-12, the most common isotope of carbon, and carbon-13 in comparison with a reference. Since during photosynthesis plants take mostly carbon-12 instead of carbon-13, the carbon isotope discrimination is calculated on the basis of the carbon isotope composition of the atmosphere. This carbon isotope discrimination means low carbon isotope composition or low carbon-13 concentration in the plant. The cleavage or formation of atomic bonds, as well as other processes affected by atomic mass, determines the change of proportion of carbon isotopes. This phenomenon is called isotope fractionation. Thus, some substances from zinc or source organs are enriched in the heavier isotope, while others become depleted. Our aims are to compare physiological performance and biomass accumulation of different genotypes along the growing period, and to test the reliability of carbon discrimination by leaflets and tubers to explain and predict the varietal differences in biomass accumulation and tuber yield under different soil-water conditions. An experiment was carried out in a glasshouse. Plants were grown in pots and maintained in water-saturated conditions until tuber initiation onset. After this period, two irrigation treatments were tested: well-water conditions, where plants were irrigated to full capacity, and water restriction conditions, where plants were irrigated at 50% of the full capacity. The water quantity for desirable soil moisture was determined gravimetrically. 3 potato varieties were tested: Desiree, a European cultivar which is considered as a drought-sensitive variety and which is common in carbon isotope studies. The other genotypes were improved varieties selected in dryland systems, namely Unica, a drought-tolerant variety, and Sarnaf, a variety with acceptable yield under heat and drought conditions. We measured stomatal conductance using a portable photosynthesis system before each of 6 sequential sampling of leaves and tubers. The samples were dried and grown using the ball mill. Grown samples were used for nitrogen content analysis and for isotope composition. For the determination of this last, the samples were enclosed in thin capsules and analyzed in a mass spectrometer. Sarnaf produced the highest tuber biomass in both watering regimes. Carbon discrimination showed significant differences between irrigation treatments. Under water restriction, black circles, less carbon discrimination occurred due to the reduction of stomatal conductance. In this figure, we can see that high carbon discrimination occurs because when the stomatal is open, the diffusion of carbon-12 is higher, and Rubisco takes preferably this isotope discriminating against carbon-13. However, when the stomatal is closed, Rubisco has no choice and takes more carbon-13, the discrimination against it being low. There are some situations in which the stomatal conductance is high but the discrimination is low. This case occurs when the activity of Rubisco is fast, fixing quickly the carbon-12 and also carbon-13. This fast Rubisco activity is related with higher photosynthetic capacity. This situation occurred under well-water conditions, as shown by Sarnaf, that had the lower leaflets carbon discrimination but the higher stomatal conductance. The higher nitrogen content per area, a trait closely related to Rubisco activity, confirmed our suggestion that Sarnaf show an improved photosynthetic machinery. However, the lower leaflets carbon discrimination value in Sarnaf was mainly observed at early developmental stages, before tuber initiation, which highlights the importance of monitoring of this trait in plants at early development stage. These results coincides with results from another field experiment in contrasting environmental conditions, which highlights the relevance and consistency of isotope assessment during early developmental stages. Under water restriction, Sarnaf and Unica showed lower leaflet carbon discrimination than Desiree. There were no significant differences among varieties in the nitrogen content per area. The higher stomatal conductance in Sarnaf and its higher biomass under drought suggests that the carbon diffusion is one of the main drivers of photosynthesis under water restriction. The lower value of carbon discrimination in zinc organs, tubers, are attributed to the mobilization of carbon to the source organs, leaves, in potato, which causes carbon fractionation. In this case, the lower value of tuber carbon discrimination in Sarnaf suggests higher mobilization of carbon to supply energy of the different tolerant mechanisms that this genotype has evidence. For example, osmotic adjustments, reactive oxygen species detoxification, maintenance of protein folding. Finally, drought-tolerant indexes have negatively correlated with leaflets and tuber carbon discrimination, which remarks the importance of assessment of these traits in drought-tolerant selection programs. In conclusion, carbon-13 discrimination allows to understand some mechanisms adopted by contrasted potato varieties. Under well-water conditions, the photosynthetic capacity is the main driver for tuber yield. This capacity must be assessed at early developmental stages, using carbon discrimination in leaflets of plants before tuber initiation onset. Under water restriction, our results suggest that carbon diffusion via stomatal or mesophyll and tolerant mechanisms are the main drivers for biomass accumulation. We recommend to assess our findings in more genetic materials of potato at early developmental stages. We recommend also that carbon-13 discrimination must be carried out before senescence in tubers. This study received the financial support from the CIR research programs CRP on climate change, agriculture and food security, CCAPS, root tubers and banana, RTV and dryland systems, and also the bilateral project BMC. I would like to thank to my co-authors José Rolando, Wendy Yactayo, Philippe Mondbu, and Roberto Quiroz, and the technical staff Nicolai Alarcón and Jesus Amayoa. This study has been recently published in Journal of Agronomy and Crop Science.